Very good, thank you.
Maryland Life Sciences would like to recognize today's supporting sponsor, Johnson & Johnson Innovation. J Labs at Washington, D.C. is a global life science and healthcare incubator network that provides early-stage innovators with access to all their needs. Located on the new Children's National Research and Innovation Campus, J Labs at Washington, D.C. also serves as the hub for Blue Night, a joint initiative between J Labs and the Biomedical Advanced Research and Development Authority. Our contributing sponsor, GSK. At GSK, we unite science, technology, and talent to get ahead of disease together. We prevent and treat disease with vaccines, specialty, and general medicines. For more information, go to gsk.com. Our keynote sponsor, DPR Construction. As the #1 pharmaceutical builder in the country for the last 5 years, DPR constructs cutting-edge spaces that challenge norms and foster discovery, allowing our heroes in lab coats and Tyvek suits to change the world. We bring integrity, enjoyment, uniqueness, and an ever-forward mindset to each engagement. DPR is ready to partner in building great things, great teams, great relationships, and great projects. We'd like to thank these and all of our sponsors for their continued support of the Maryland Life Sciences Bioinnovation Conference.
At Novavax, we are relentlessly committed to one goal: providing protection against the world's most urgent infectious disease threats. Protection for families, for communities, for the world. We are working to create best-in-class vaccines by focusing on what matters most: proving safety, establishing efficacy, trusted science, persistent hope. A protein-based established formulation is our foundation. An innovative recombinant nanoparticle technology platform is our difference. Developing adjuvanted vaccines against emerging and evolving infectious diseases is our strength. Our team of experts is working in partnership with leading organizations from around the globe to resolutely advance our pipeline so that whenever a vaccine is needed by anyone, Novavax is there. This is our commitment. Novavax.
Our next panel will discuss emerging vaccine opportunities and modulating the immune system. Please welcome your moderator, Sally Mossman, Vice President, Head of Research Portfolio and Strategy, Sanofi.
Hello everyone, and thank you for sticking around despite the fire alarm for this next panel session, which I'm sure you'll agree is going to be very exciting. We've got some wonderful panelists gathered together for you today from different backgrounds. We've got industry, CRO, and then a couple of, an academic as well. So really looking forward to the conversation. So I'm not going to introduce the guests. I'm going to rather ask them to do that themselves. So let's start with our virtual participants. Chris, let's start with you.
Hi, everyone. Sorry I can't be there in person. I'm Chris Jewell. I'm a professor of bioengineering at the University of Maryland, College Park. and my lab does a lot of work at the interface of material science and immunology, specifically on antigen-specific immunotherapies. Happy to be here.
And Chris, you're local, but currently joining us from London, right?
Yeah, exactly. Sorry, I had another speaking engagement, but I wish I could be there, especially for the cocktail hour.
Jeff, let's move to you next.
Yeah, hi everyone. Pleasure to join you. Wish I could also be there in person. I'm Jeffrey Lynn, and I'm the Senior Vice President at Vaxitech, heading up the Synthetic Immunotherapies Division. My background is actually in synthetic chemistry, and I coupled that with cellular immunology training at the Vaccine Research Center at NIH, working with Bob Cedar on different T-cell-based immunotherapies, and also did my medical training at Johns Hopkins. So have been trying to spend most of my life thinking about how do we use synthetic approaches to design kind of next generation of immunotherapy. So eager to join this discussion. Thank you.
Fabulous. Thanks, Jeff. And Tim?
Hi, everybody. My name's Tim Clay. I'm a Vice President of Discovery at GSK. My group works on targeted immunotherapies, so really looking to develop therapeutics really for chronic disorders. My background is in tumor immunology, but I've got extensive experience in vaccines and cell and gene therapy R&D. I'm looking forward to the discussion.
Super. And last and definitely not least, David.
Yeah, thank you. My name is David Pepperell. I'm a pharmacologist and toxicologist with Biologics Consulting based in Alexandria, Virginia. I've got a background in cell molecular biology as well as pharmacology and toxicology, and help folks consult on development, early stage product development at biologics. So I'm happy to be here.
Great, thanks everyone. So I think having this panel coming right after a COVID vaccine session is really a nice juxtaposition in terms of the first question that I'm going to ask. And Tim, I'm going to direct this at you. But of course, everyone jump in if you have something to add. So we're all very familiar with prophylactic vaccines, a tremendous track record in terms of successfully combating infectious diseases. But if we're talking about therapeutic vaccines, could you talk us through some of the specific challenges and the obstacles that need to be overcome in that setting? What are some of the key differences there?
Yeah, I think obviously everybody's very familiar with prophylactic vaccines. You generate an immune response that protects a person against a future potential infection. In the therapeutic space, we're really not dealing with a healthy person. We're dealing with a patient who already has disease. They may actually have, say, HIV viral reservoirs, so they have persistent infection. They may have an autoimmune disease, so they actually have an immune system that's actually attacking them. They could actually have cancer as well. So we're looking at a completely different sort of paradigm in terms of the way we're approaching this. We're looking at patients who have disease, and therefore quite often their immune system is actually affected by the disease. It's impacted. It may be immune dysfunction in the case of autoimmune disease. In cancer patients, there's a lot of immunosuppression from the tumor, which means that there's sort of T-cell exhaustion and the immune system's not able to function correctly. So when we talk about actually developing an approach of immune modulation or modulating the immune system in that setting, we're going up against not a normal immune system, but one that may already be sort of subverted. And I think a key aspect of it is also, as we think about developing these things, patients are actually already on a therapy quite often, and that therapy may also be modulating their immune system. In the case of cancer, patients are getting immune checkpoint blockade, immune checkpoint inhibitors. And so we'd be looking to actually produce a vaccination on top of that that's actually going to augment the activity or synergize with an existing product. So it's a very different paradigm when you think about a therapeutic vaccine. You have to take into account the biology of the disease. And then I think lastly, I would say there's also a very different regulatory path. So we're very used to prophylactic vaccines. The regulatory path is very clear. When we talk about a therapeutic vaccine, actually the therapeutic vaccines will probably go down a different regulatory path. These are actually disease-modifying therapies. They're not preventative therapies. So I think that's an area where actually there isn't really a proven path for the development of these approaches. And I think as a sort of industry collective with biotechs and also with regulators, we actually have to have a dialogue on actually how to develop these therapeutic vaccine approaches, because there isn't really a path that's well-worn yet.
So hold that thought, because David, I'm going to come back to you in a minute on the regulatory question, because I know you have a lot of experience in the regulatory setting. But first, Chris, I'd like to ask the question a little bit differently to you, which is, what are some of the key learnings that we can take from prophylactic vaccines and apply to the therapeutic setting?
Yeah, I think building on what Tim said, I mean, you have a patient with potentially with a disease. They may have pre-existing immune causes because of that, or they may have because of a drug they're taking. So I think from the design perspective, you kind of think about what are the key parameters of a prophylactic vaccine. I mean, you have memory, that's obviously unique among other therapeutics. You have the target, and then you have, sort of the plasticity. The T cell plasticity, instead of just giving a drug or a monoclonal like we might do in some of the existing therapies to block or upregulate a pathway, we actually want to think about how do we generate the types of phenotypes. So if you have effector cells during autoimmunity driving disease, could you expand regulatory cells that control those cells? Could you even maybe convert the existing inflammatory cells to regulatory cells? So transdifferentiation is a really hot area. In oncology, you probably want to undo that. You may have T cells that are antigen-specific, tumor-specific, that are suppressive or inhibiting the normal aspects of immunity. So could you do something to address that? So first of all, thinking about T cells as a major target, not as a druggable target, but as something you actually want to expand and modulate. And so that brings us to the target. I mean, in prophylactic vaccines, we're trying to get things to lymph nodes or spleens or other immune sites. So instead of designing things to go to a tumor, how do you actually condition a local environment to make it accessible to generate the right kinds of responses? And there are also lots of other neat tissue-specific effects emerging in prophylactic vaccines that could be very useful in cancer and autoimmunity. You've got tissue-specific resident T cells, so cells that have encountered an antigen, and we now know these small populations of resident memory T cells don't need re-encounter with an antigen to have an effect. That could be very useful in tumors where maybe there's a window of accessibility because of, as Tim said, because of the combination therapies that patients might be on. If you have autoimmune disease, you've got a specific site of disease that's pathology in the immune system is driving, maybe the CNS in multiple sclerosis, islets in type 1 diabetes, joints, so the target. And then lastly, depending on how speculative we want to get here, but memory, right? This is a key component of prophylactic vaccines. So could we generate durable effects, both to take advantage of the increased potency that comes from memory and the durability that comes from memory? And so in immuno-oncology, that'd be nice for increasing the outcomes of your immunotherapy, and also maybe for minimizing relapse. And likewise, during tolerance, if we're trying to expand regulatory T cells that could go and in an antigen-specific way control existing populations, or maybe make Tregs that have durable phenotypes or survival markers. So, some might call that regulatory memory. And if you want to call it that, or something safer like durable tolerance, either way, there's a lot of great immunology being done that can be exploited and is starting to be exploited for antigen-specific immunotherapies, like in cancer and autoimmunity. And I think some of the parameters targeting innate receptors to direct the downstream outcome of these T cells, and also valency, like what are the relevant antigen repertoires, what antigens should we give? So these are all things that we as the field have done a lot in prophylactic vaccines, and I think there's some really cool opportunities to exploit these going forward in immunotherapy. So I'm looking forward to further discussion.
Oh, fabulous, Chris. And I think your answer really highlighted, I think, the need for this really deep science around the immune system, but also how innovation maybe can drive some really, truly groundbreaking approaches here. You touched on some of the indications that could be addressed through therapeutic antigen-specific vaccine approaches. Jeff, would you like to kind of expand on that a little bit for us? Oh, yep. I think you're on mute.
Yep, okay. Absolutely. So I think I agree with everything that Tim and Chris mentioned. I would say probably the way I think about earlier generation vaccines and where we hope to see the field go is really delineating between prophylactic vaccines as being antibody-mediated immunity And then the kind of next generation of vaccines really leveraging more of T-cell mediated immunity. And a lot of the basic science discoveries over the last couple of decades have tried to really understand how do we generate different different T-cell populations, so CD8 T-cells, those cytotoxic T-cells for cancer and treating chronic viral infections. And as Chris mentioned, how do we induce regulatory T-cells for tolerance for treating different inflammatory diseases? So really, it's those discoveries that I think are paving the way for groups like many of the folks on the in this conversation today for developing therapies for different applications. So in terms of the fields, really, I would say there's probably, at least how we're thinking about it, Vaccitech and within other companies, we're really thinking about a couple really big opportunities. In the oncology space, I agree with Tim. I mean, the big challenge there is you have a host that's living with a chronic disease. They often times have a exhausted immune system, and they oftentimes even have pre-existing T-cell responses. And so I think a lot of the audience is probably well aware of the advances that have been enabled by checkpoint therapies, including things like drugs like pembrolizumab and nivolumab, as well as ipilimumab. And so these checkpoint therapies allow you to reverse some of the suppressive mechanisms of cancer and allow T cells to be unleashed and mediate tumor killing. However, we know that a lot of patients don't have pre-existing T cell responses in oncology, and we know that the tumor can be a very suppressive environment. So even if you have T cells plus adoptive cell therapies, how do you create a permissive environment for killing? And so one of the big opportunities we see for immunotherapies in oncology is combination immunotherapies. So we know that we can provide T cells either through vaccination or through adoptive cell therapy, and then that those can be combined with checkpoint inhibitors to unleash their killing. However, some of the latest advances suggest that we can actually modulate the tumor microenvironment to allow those T cells to permit killing, and then can even use combination of adoptive cell therapy with vaccination. For example, giving T cells, but then boosting those T cells that are transferred over through vaccination allows you to continue to maintain the high magnitude T cell response that you need to mediate killing. So really in the oncology space, there's a lot of good technologies already out there. And one of the questions is how do we best combine those to really systematically address all the challenges that we have for fighting cancer. The other really big opportunity that I know a lot of folks in the immunotherapy space are particularly excited about is leveraging new knowledge about how we can induce regulatory T cells, or as Chris indicated, how can we take pro-inflammatory cells and actually trans-differentiate them. So take a pro-inflammatory CD4 T cell and actually convert it to a regulatory T cell that mediates tolerance. So this is a huge new opportunity. And so a lot of folks our group at Vaccitech are applying the knowledge that we have for inducing T cell immunity for cancer and for infectious diseases, and actually now leveraging that for inducing regulatory T cells for treating autoimmune diseases. And I'll just be brief here and say that when I say autoimmunity, I really mean to say inflammatory diseases, because when we talk about regulatory T cells, there is an opportunity to develop curative therapies. So these aren't just disease modifiers. These are potentially curative therapies for things like allergies, various autoimmune diseases, including, for example, multiple sclerosis, type 1 diabetes, inflammatory bowel disease, as well as transplant rejection. I would also add that there's a number of other diseases, including even atherosclerosis, that is actually probably largely mediated by inflammation to oxidize lipids. So really, there's a huge opportunity to try to leverage immunotherapies that can induce regulatory T cells for treating these inflammatory diseases, and see a tremendous opportunity there. So I'll stop there. Thanks.
Yeah, thanks, Jeff. So very broad, and as you say, tremendous opportunities if we can really address some of the key challenges around the redirecting the immune response. So moving on from regulatory T cells, let's talk about regulatory authorities, David. So can you talk to us a little bit about the question that Tim actually raised around which, CBER versus CDER, how would these types of products be regulated from your perspective?
Well, certainly a lot of it hinges on the nature of the product itself and what its aim is. Again, there's certainly prophylactic vaccines, there's therapeutic vaccines, and they're regulated differently within the FDA, both within the Center for Biologics, CBER. So the traditional preventive vaccines we're mainly all familiar with, and we've all probably had multiple doses over the last several years, are regulated by Office of Vaccines, OVRR. And as Tim mentioned, the requirements for vaccinating a healthy population, as even young children and babies, versus vaccinating or treating late-stage cancer patients perhaps are very different. So the requirements are different, and the paradigms of delivery of the products are very different. So they have different mandates essentially to show safety in a healthy population versus therapy against maybe a specific tumor or even a patient-specific tumor in the case of therapeutic vaccines. So actually these products are regulated by totally separate entities within CBER, either Office of Vaccines or Cell and Gene Therapy, Office of Tissue and Advanced Therapies, or OTAT. So the Office of Tissue and Advanced Therapies, which also regulates cell and gene therapies, adenoviral vectors, stem cells, any cellular therapy or live products, will typically regulate the therapeutic vaccines, even if they're not necessarily cellular products, versus a preventive vaccine will be going to the Office OVRR, which, again, the requirements are a bit different, and it really dictates the nature of the product will drive where it's regulated and what the expectations are. And of course, your patient populations are different too, either the healthy population in the case of preventive or potentially very ill patients, and the levels of risk you have there is what really drives the program, the non-clinical program at least.
Mm-hmm. And are we seeing sort of collaboration between the two divisions in terms of leveraging the expertise that sits in one versus the other?
Well, usually if it's one product is designated toward a specific office, they will take the lead. And this also goes toward things like combination products too, where you may be dealing with a drug and a biologic. They will consult with each other. But one certainly takes the lead. So if you have a therapeutic vaccine, it's going to go to OTAT, Tissue and Advanced Therapies. That will really drive the expectations of like what's, at least from a non-clinical perspective, what studies you have to do, looking at the clinical population, and again, bearing in mind how sick they are and what the level of risk is versus a healthy population that's never seen this product before, and millions of people will be exposed to it. So there needs to be a level of cooperation. I don't think it's too routine that within Center for Biologics that they will collaborate routinely, but the expertise is there for them to leverage.
Yeah, super, thank you. All right, Chris, I'm going to turn to you again now, and would love to hear a little bit more from you about some of the synthetic approaches and nanotechnologies in particular that you're interested in pursuing within this space.
Yeah, I think, and I'm sure Jeff will probably talk about some specific examples, so maybe I can kind of give some orientation for folks that might not be as familiar with materials and nanotechnology. So, I mean, everyone probably knows nanoparticles now, if they didn't before, because the COVID vaccine, of course, is formulated in lipid-based RNA vaccine as a nanoparticle. So I would kind of think about it like 3 scales. So you have nanoscale materials, whether these are lipid or polymer particles or some kind of other type of non-viral vector. And you might use these to deliver antigens, a tumor antigen or milieu and adjuvants with regulatory cues. And nanomaterials have a lot of special features to enable these types of things. For example, you can control the valency or the density of signals. You can ensure immune cells or tissues encounter specific combinations of cues. And these, of course, are all things that have great implications for the immune system, where it's an integration of both the signals present, the concentration, the duration. So at the nanoscale, basically the opportunity to incorporate different classes of materials, of immune cues, and perhaps deliver antigens with an adjuvant for cancer immunotherapy or antigens with regulatory cues to redirect how those antigens are responded to. So I think one thing this means is generally having good knowledge or as best information as possible about the antigens driving disease in some of these inflammatory conditions, and also obviously in terms of tumors. At the micro scale, there's a lot of interesting work, for example, making depots that are loaded with cues that could be implanted, and some of this is now in clinical trials, to condition APCs that come in, so recruit the antigen-presenting cells, give them some cues that would be useful, and then send them back to lymph nodes to initiate responses. Something we're working on is actually scaffolds that locally condition the lymph node environment so that we make the cells we want in that site, but we don't have to systemically expose, in most of our work preclinical, although now we're starting to translate some of this, but you don't have to worry about exposing the potent immune cues throughout other sites, just using the lymph node like a factory. So that's the micro scale. Then of course, devices at the sort of macro scale. One cool example are microneedles. Many folks are probably familiar with these. They're short polymer or metal needles that are long enough to target skin-resident immune cells, but too short to hit pain receptors. So there's a couple approved indications and some high-profile trials for like flu. And because patients can potentially self-administer these, and they don't need cold chain, and so there's a lot of interest in prophylactic vaccines. But one thing a lot of folks are interested in for immunotherapy is that these are very efficient at targeting the APCs in the skin, and APCs in the skin are privileged and have overexpress a lot of different innate inflammatory pathways, and so it's also sort of a very efficient way to try and drive therapeutic responses. So that's kind of the scales I would think about with materials. And then the other thing I'll just plug is materials are also tools to study immune processes, even if you don't want to use a nanotechnology or a particle or something later. So for example, because you can display cues at certain densities or certain combinations, you can really study something about, maybe how do you engage a TCR? There's some beautiful work by Perry Santamaria's lab where they took MHCs and displayed antigen recombinantly, and then they let those engage T cells, and they really dissected the effects that has on T cell receptor formation and how that drives different immune outcomes. We've done some work displaying immune ligands on surfaces and then using microfluidics to measure binding constants. So I think that's also a really neat emerging area, using materials as tools. And then of course, everyone realized the potential of coupling this kind of stuff with big data, AI, immunometabolism. So I think, I think of it as therapeutics and also opportunities to study processes.
Very exciting. Thanks, Chris. And Jeff, I noticed you vigorously nodding your head while Chris was talking, so I'm going to give you an opportunity to jump in here as well.
Yeah, I'm usually vigorously nodding my head when Chris is talking because he's got a lot of good stuff to say, just like our other speakers up there. So, yeah, so I think one of the things that we talked about before is an extension of nanotechnologies are what we refer to as synthetic immunotherapies. So Chris mentioned the RNA vaccine as a lipid nanoparticle formulation of RNA. Arguably, that's kind of a semi-synthetic approach because there's a combination of synthetic with recombinant. And so I think probably a question for a broader audience like this is why make this distinction between synthetic immunotherapies and other types of immunotherapies? And I think it's important when we talk about opportunities to really describe what new technologies allow us to do. And so if you look at what I'd refer to as conventional immunotherapies that are really based on recombinant technologies like recombinant proteins, viral vectors, whole pathogen, these kind of technologies are really, have been tremendously advantageous to society, but they have their intrinsic limitations. And I'll give a couple examples and why we've gotten into this area of synthetic immunotherapies as a complement to recombinant approaches. And so one of the examples is, for example, just as an example here, a lot of cancers will highly express different types of sugar molecules on their surfaces. This is also true of different viruses. There's a number of different sugar molecules that are highly represented on the surface of different viral particles, including SARS-CoV-2. Our group initially got into this at the Vaccine Research Center because we wanted to go after targets on the surface of HIV virions that had a lot of glycosylation sites. Now, the challenge with recombinant technologies is to go after some of these targets through vaccination. You don't always have control over, these sugar molecules. And so there was a need to actually make vaccines that have these sugar molecules that had to rely on technologies other than recombinant technology. And so we can actually, using synthetic approaches, we can go after a lot of different potentially targetable antigens that are just not possible with existing recombinant technologies. And one example that we're going after in cancer is trying to target glycoproteins that are highly upregulated in different cancer tissues. And this is something that you currently have to access synthetically by actually building the vaccine, compound by molecule by molecule, and that you can't currently encode genetically. And so that's one of the advantages, or one of the areas of differentiation, I would say. Another really kind of interesting thing with synthetic immunotherapies is that if you imagine you want to induce tolerance in someone, if you use a lot of our traditional vaccine technologies, a lot of these are pro-inflammatory. So all of us who've gotten the recent vaccines, I mean, we notice there's a little bit of inflammation at the site. It gives you a little bit of soreness, but that's the vaccine working. But for tolerance, you don't necessarily want a pro-inflammatory environment. And so one way where we can precisely program the type of immune response this induces is by building the vaccine, from the ground up. And so Chris mentioned different nanoparticle technologies. A lot of these nanoparticles are inherently non-inflammatory. And so what you do is you take those components and you build a vaccine basically from scratch using the synthetic approach. So you can have a nanoparticle, it can deliver an antigen that you want to induce tolerance against, and then you can also co-deliver different immunomodulators to try to create a purely suppressive, pro-tolerogenic environment rather than a pro-inflammatory environment. And so that's really the big distinction I would make when we talk about novel immunotherapies. I think some of the novel immunotherapies that are, at least we're really excited about, are synthetic and semi-synthetic approaches. And the reason why we're excited about these is because they give us access to new targets, like I mentioned, new targets in cancer, new targets in infectious disease, and they potentially give us access to whole new therapeutic areas, including tolerance, where you may need a platform that's inherently non-immunogenic, which is not something you necessarily have with a lot of our earlier generation immunotherapy and vaccine platforms. So I just wanted to make that distinction as we think about in this session, what are the kind of new opportunities in emerging immunotherapies. I'd make the distinction that really I'd break them down into synthetic, semi-synthetic, and recombinant.
Yeah, thank you. And there's been some recent work as well showing that even mRNA, through some sequence adjustments, modifications, et cetera, can be rendered more tolerogenic. And those, we all know, right, that actually the current form of the pandemic vaccine is quite inflammatory, and that's why we sometimes spend the next day in bed after vaccination. But it's working, as you say. But so, David, back to you. You do a lot of work with various partners around non-clinical development of therapeutic vaccines and other approaches. Could you talk us through some of the considerations from a non-clinical perspective?
Well, certainly, I think the greatest one is understanding that, certainly the animal models that we use are not human, and the systems are very different, and you're trying to, elicit immune response in a very different system. So, over the years for prophylactic vaccines, it was pretty straightforward. You'd have an antigen and mix it with an adjuvant or alum or something like that, and inject it into a rabbit, and you get antibodies and get the vaccine approved or generated in chicken eggs for flu vaccine. And but now with those are, but those the bad actor here is a foreign, a bacteria or a virus. So that's a foreign proteins that we're targeting. Now with a lot of these newer products in oncology, I mean, they're potentially human proteins or mutated human proteins, so they're itself essentially. So the challenge is to often find the right animal model or one that's relevant. And if you've got, say, if you're studying a human protein in an animal, that's immediately going to be immunogenic unless maybe you go into a non-human primate. But even then, it's never a perfect model. So I think the key challenge that we have in non-clinical is finding the right animal model, finding something that's, the FDA would at least say relevant, biologically relevant, or, and then having a combination of in vivo studies as well as in vitro studies, possibly with human cells, human tissues that you can leverage and put together a sound package. But when it comes down to it, is really understanding all the immunology that everybody has been, speaking to. It's very complex. It's only getting. It seems like it's only getting more complicated. Not being an immunologist, and then pulling that together and putting a good package together for regulators to say, this is what we know. This is why we think it's safe. This is why we think it works. I think that's really if you can find the right animal model and understand what the product is and how the product is working, that will drive the nature of the scope of the non-clinical program.
And in a setting where the vaccine or the immunotherapeutic is going into patients, I suppose there may be a little bit less burden in terms of that preclinical piece, right? Because, in fact, the only way to really address the question is by going into a patient population. And of course, the risk-benefit equation is different compared to a prophylactic.
Ultimately, I mean, if it's already been in people or something, a very closely related product, you can often leverage safety. But if it's still a different novel product, there generally has to be some non-clinical work. And if there is a relevant animal model, that's fine. You can leverage that. If it's a monkey or if it's a syngeneic mouse model, that works great many times. But they're not perfect. The immune systems are different. You have to bear that in mind, and regulators understand that. So, but I think it's if you know going in what the target is and what the issues are, you can design or come up with a relevant model. And usually that comes from the basic science, and that's how we at least think through what the basic safety concerns are and how we then design the efficacy studies, immunology studies, and then pair that with the safety like GLP tox, and hopefully there'll be something that'll be relevant that will be able to address those questions.
Yeah, super, thank you. So Tim, I'm going to come back to you again. I know earlier in your career you have been very much involved in cancer vaccine programs. So many approaches have been tested clinically, many have failed over the years.
Yep.
What's your kind of expert opinion around, where is the field now? What do you think is the probability of success? Is this still something we should endeavor to be developing?
Yeah, it's a great question, and I'll probably date myself here, but since the early 90s, I've worked in this sort of tumor immunology space. And there hasn't really been a successful cancer vaccine since then. But I think as Chris and Jeff have already touched on, there's actually a lot of reasons to believe the immune system can actually treat cancer. So obviously, chimeric antigen receptor T cells are actually a real breakthrough in hematologic malignancies. I think as Jeff mentioned, immune checkpoint inhibitors are actually activating T cells that are present in patients and actually causing them to reject their tumors. Usually at about 10, 20, 30% rather than all patients, so the response rate's not perfect by any means, but there's definitely evidence the immune system can actually clear tumors. And I think actually in the course of that sort of 20 or 30 years, partly with the development of these kind of therapies that are working, we've learned a huge amount about how the immune system tackles cancer. So I feel like we're in a position now where we actually understand why a lot of those previous iterations of vaccines didn't work. I can give you many different examples, but some of the key ones are quite often, people used a single antigen if you elicited an immune response, you'd actually kill tumor cells expressing that antigen, but then the ones that didn't would grow and replace them. So you'd actually select for a tumor that evaded your own therapy. So it's very clear we need to have more than one antigen. We also need to have a very high-quality immune response. We understand a lot more about what features we need. And so not just CD4, but a very strong and potent CD8 response. We now have technologies that allow us to actually get really strong CD8 responses. Some of the previous technologies that were used were not good at that, but things like RNA and viral vectors give you very strong immune responses that are CD8 as well. You can encode multiple antigens in them too, so the technology platforms have certainly advanced. I think the other aspect that's really important and probably was not recognized, certainly in the distant past, was that you actually need to combine this with other therapies. And so the immune checkpoint inhibitors really give us a window where we're seeing responses in patients, 20 or 30%. If we can add to the T cells that are produced with vaccination, maybe we can bring this up to 50 or 60%. So we can actually really see an opportunity to synergize. And then I think lastly, I would say in terms of patient selection, I think we've advanced enormously as well. And so we need to look for patients who actually have an immune-infiltrated tumor, what we call a hot tumor, where the immune system's already there, but it needs to be reactivated. That means the T cells can get into the tumor versus patients who have an immune desert in their tumor where there's just no immune cells getting in. We also need to look for tumors where there's actually a high mutational load. There's a lot of mutations in the tumor, means there's more potential targets for the immune response. And actually some of those mutated targets, which we call sort of cancer neoantigens, may be the best targets for these kind of approaches too. So there's been a huge sort of development in terms of understanding. So after 20 or 30 years in the field, I feel like we're poised, hopefully, to see a success. And I kind of think back, I mean, part of the work I've done in the past is in gene therapy. Huge hype 20 or 30 years ago, and it took 20 or 30 years for gene therapy to be successful. Even last week in the news, you saw there's an Alzheimer's drug that actually seems to actually be having some efficacy in patients after a huge number of failures in the Alzheimer's space. So, I'm in discovery. We have to be optimistic. We're trying to push the boundaries. But I feel like, looking back on my career, Sally, I'd kind of say I think we're actually poised to see some real opportunities for things to work there. So I'm encouraged, actually.
Love it. That is a great note to end it on, because I do want to leave enough time for maybe one or two questions. We haven't got much time, but please, if you're in the audience, please step up to the mic. And while you're doing that, I just want to thank the panel for a really interesting and inspiring discussion, actually. Very much appreciate all of the insights that you've shared with us today. So, question, please.
Yes. Hi, I'm Mark Shennerman from UMBC. I didn't hear anyone say anything about mucosal immunity. So what sort of innovations are in the works to increase mucosal immunity in patients, particularly to address the unmet need for infectious diseases?
Who wants to take that one? Anything being done for me? Yeah, go ahead, Jeff. Go ahead.
Yeah, it's a great question. It's one that I think every time we have a discussion, particularly about novel approaches for targeting respiratory viruses, we're always talking about mucosal immunity, right? Because that's the most important place to get your IgA antibodies is being produced in the mucosa. And so I would say at least some of the innovations that we've seen in that space, some of the thinking that we've had working at the Vaccine Research Center and then with some of the colleagues at Vaccitech is that the route of administration seems to be very important. So it might be as simple as taking existing vaccines for respiratory viruses and actually providing those by intranasal route of administration. Or it might require redesign of some of those platforms to allow them to be more tolerated by the intranasal route or to allow for better expression. So if it's an expression system like RNA, one of the questions is, do you actually get good RNA expression by the intranasal route versus the intramuscular route, which is the current route of administration? And so at least those are some of the things that we've come across in the space. But Chris mentioned some of the patches. I think some of those technologies have also been looked at for, can you program the type of immunity that you're getting through other routes of administration, in addition to intranasal.
Yeah, maybe I can just briefly add, in addition, I mean, there's a lot of work to try and explore how to confer different homing markers onto the cells that are generated. And so some of that is through activating innate pathways. Some of that has to do with chemokines during differentiation. So I think that's another opportunity, not just in actual immunity, but to direct the fate, the geographic fate of those cells. And of course, there are tertiary lymph node organs that have higher level structure that might be additional targets that, just like in prophylactic vaccines, these play important roles in generating the immunotherapeutic effects when you have an antigen-specific presentation. So I also agree with what Jeff said.
Great, thank you. We do have one more question, so let's make it a quick one. Thank you.
Sure, yeah, quick question for the regulatory side, David. I promised you I'd have a question. You mentioned finding the most relevant animal model. So are we going to outdo ourselves with technology, with all these nanoparticle therapies and synthetic this and synthetic that? I'm always interested in the public view. So a lot of the public felt like our mRNA vaccines skipped animal studies. And so are we going to outdo ourselves here and not have any more relevant animal models in the future sooner than later?
Yeah, that's a great point, Angela. And that the funny thing is that, years ago, biologics would be considered rather, dirty. They'd just be unpurified, fractionated blood or dirty cell products or very complex biologics with a lot going on. But now they're getting so specific and so even patient-specific for autologous tumor therapies or patient-specific tumor therapies. In a sense, yeah. I mean, if you can't find a relevant animal model, you can't study the product. If it doesn't give you a meaningful immune response or it doesn't respond in a way that mimics the human scenario, you're not going to learn anything. So I was thinking of it, and I was thinking about this session, that these products are like a double-edged sword. If they get more specific, and that's great, so they'll be safer, in theory, for the patient. There may be very few risks if they're taking their own cells and inserting a gene that's well-known and delivering it back to that same patient. But then you need some safety data on that, and how do you test that in animals if it's human cells in an animal model? Especially with the immune system, you can't just test in immunocompromised animals. You need to use animals with a functioning immune system. So it really makes things complicated as the products get more honed and more precise, that it's harder for us non-clinical people to study them, and we have to come up with better and better animal models or leverage in vitro systems when we can to in vitro human cells to satisfy regulators, because we'll always need some data to get into the clinic.
Well, thank you, and we'll have to end it there, but thanks very much to the panel.